Hello everyone and welcome to our lecture focused on the concept of gene therapy. Today we are going to be talking a bit about how to hack the human genome and how to deliver those newly constructed gene variants. In order to understand the concept, we need to go back to the introduction to genetics lecture that I delivered at the very beginning of the academic year. Over there we have learned that genetic disorders cannot be cured by a pharmaceutical intervention. So the only thing we can do conventionally is to treat the symptoms and basically possibly slow down or halt the progression of the disorders. However, nowadays many technologies have emerged, including stem cells, but also knowledge about the human genome. So in order to understand today's lecture series, we need to go back and recapitulate what we have learned about stem cells. So stem cells are cells that can either replicate themselves, generate identical copies, or specialize or differentiate into many different cell types, including cell types such as neurons, blood cells, muscle cells, and so on. Regarding the stem cell types, there are two major cell types that we need to be aware of. The first type being embryonic stem cells, which are found in the embryo. They can form almost any cell type in the human body and they have very high division rate. The second type of stem cells we need to remember are adult stem cells, sometimes referred to as tissue-specific stem cells. Those cells are present in nearly all tissue types, and under physiological circumstances, those cells are quiescent or dormant, so they are inactive. However, after injury or if there is a degenerative disorder going on, those cells can re-enter the cell cycle, proliferate, and contribute to regeneration. The third type of cells that we need to keep in mind are so-called induced pluripotent stem cells, and those are artificial pluripotent cells generated from cells isolated from the body. So they are generated artificially in the lab, and all of those cell types can be used within gene therapy. Again, in the recent years, many different cell therapies and gene therapies have actually been approved by many regulatory bodies, including the US FDA. So this is already clinical reality. The next thing we need to keep in mind if we want to understand gene therapy is the nature of certain mutations that we have learned within our genetics lecture series. Here, the first type of mutation that we need to remember is addition. Here an extra base is inserted into the DNA code. Therefore, all bases following the insertions are affected, and the new gene likely no longer will make sense. Again, so what I have shown you in a dedicated lecture was this example. Here we have an artificial triplet code, and this sentence makes perfectly sense. Sam met Batman. However, if we now add one base, represented by the letter L, then the whole reading frame shifts and all of the following triplets do no longer make sense. Next type of mutation that we need to be aware of are deletions. They are quite similar to additions, but here instead of addition, a base is specifically deleted from the DNA course. Therefore, similar to an addition, all bases that follow this triplet will be affected, and therefore the new gene likely no longer will make sense in the context. Again, if we use this example of the artificial triplet code, if we now delete one single letter here, then the reading frame shifts, but this time to the left hand side, and all of the following triplets are no longer making any sense. The third type of mutation that we need to remember are point mutations. Here, if the sentence makes sense, the base could be substituted and the following sequence could still make sense, right? And since our genetic code is degenerated, there are many permutations that we could imagine. So again, the new gene may still make sense, but the function might be totally different. If this mutation actually affects genes that are responsible for control, for example, of cell division, this could lead to tumorigenic transformation of a cell. We could have generation of cancer cell since those crucial genes could be affected. And one type of point mutations I've been talking a lot about is a mutation that is contributing to development of sickle cell anemia. This type of disorder is caused by a point mutation in the hemoglobin gene, where one single base at a certain position in the gene is actually affected. Therefore, the resulting protein, the resulting hemoglobin is different, and it can no longer fulfill its original function. As a result, the red blood cells change shape, are also less efficient in transplanting